Hey guys, welcome to my channel. My name is Keika. I'll be going over textbooks, chapter by chapter, section by section. And as you can see on my screen here, this is my plan for everything that I want to go over in the time span of this channel. So I want to go over biology, chemistry, microbiology, anatomy and physiology, nursing fundamentals, nursing assessment, medical surgical nursing, psychiatric nursing, geriatric nursing, which is the elderly, labor and delivery nursing, and pediatrics. And I know this looks like a lot, but as someone who's been through about 95% of nursing school, I can say this is doable stuff as long as you take the time to kind of go over the material. And that's exactly why I'm here. Like, I'm going to be reading the text and sharing my notes about the text. I think this information is really important for making lifestyle decisions and health-based related decisions. And I just hope this helps. I hope this helps students that are preparing for classes. I hope this helps students that are in classes. I hope this inspires people who aren't in school but want to go back to school to just get started on learning. And we're going to be starting with biology. This is my APA citation. We're going to be going over Campbell Biology by Yuri, Kane, Wasserman, Minorski, and Orr. This is the 12th edition book, and it came out in 2020. And I believe they haven't come out with a new edition yet, because this is the textbook that I got off their website, pearsonplus.com. I believe it's pearsonplus.com. Let me see. Pearson.com. Okay. So yeah, let's get started. Okay? So chapter 1. Evolution, the themes of biology, and scientific inquiry. And here I wrote down the key concepts. The study of life reveals unifying themes. The core theme, evolution accounts for the unity and diversity of life. In studying nature, scientists form and test hypotheses. Science benefits from a cooperative approach and diverse viewpoints. Okay, so these are our key concepts. I'm going to go over each key concept with you guys in the next series of this chapter, because I can't get through the entire chapter in one video, unfortunately. I'm going to go over the key concepts with you. And we have our learning objectives here, which I'll scroll down to. Use examples to illustrate each theme of this book. Summarize how evolution accounts for the unity and diversity of life. Discuss the scientific process. Evaluate the contribution of diversity among scientists to the scientific process, or to scientific progress. Let me edit that real quick. Progress. Okay. So, concept 1.1. The study of life reveals unifying themes. At the most fundamental level, we may ask, what is life? Even a child realizes that a dog or a plant is alive, while a rock or a car is not. Yet the phenomenon we call life defies a simple definition. We recognize life by what living things do. Figure 1.2 highlights some of the properties and processes we associate with life. So we're going to look at the figure 1.2. So the characteristics that define life are order, evolutionary adaptation, regulation like blood flow, temperature, body temperature, and digestion, energy processing, growth and development, response to the environment, and reproduction. Biology, the study of life, is a subject of enormous scope, and exciting new biological discoveries are being made every day. How can you organize into a comprehensible framework all of the information you'll encounter as you study biology? Focusing on a few big ideas will help. Here are 5 unifying themes, ways of thinking about life that will still be useful decades from now. Organization, information, energy and matter, interactions, evolution. In this section and the next, we'll briefly explore each theme. So here in my notes I wrote, biology is the study of life. 5 unifying themes for biology include organization, information, energy and matter, interactions, and evolution. And I put some stuff in parentheses, but we're gonna go over that in this video. Theme. New properties emerge at successive levels. Theme. Theme. New properties emerge at successive levels of biological organization. Organization. The study of life on Earth extends from the microscopic scale of the molecules and cells that make up organisms to the global scale of the entire living planet. As biologists, we can divide this enormous range into different levels of biological organization. In figure 1.3, we zoom in from space to take a closer and closer look at life in a mountain meadow. This journey, depicted as a series of numbered steps, highlights the hierarchy of biological organization. And here we're going to go over figure 1.3. So it goes from the biosphere, ecosystems, communities, populations, organisms, organs, tissues, cells, organelles, molecules, and they should have added the atom, but I think they'll get to that later. Okay. So, for the sake of this video, I'm not going to read all of this, but I'll summarize what I wrote here. The biosphere is a planet that consists of all living organisms on it and places where life lives. Life exists. Ecosystem, a particular area with living and non-living things, such as a meadow, tropical forest, grassland, desert, or coral reef. Communities, living organisms that live within a particular ecosystem, the set of populations that inhabit a particular area. Populations, all the individuals of a species living within the bounds of an area. Organisms, individual within the population. Animal, fungus, bacterium. So an organism is one part of the whole. Organism to population, one part, whole. Organs, a part of the organism that has specific functions to maintain the organism. Tissues, a group of cells that work together for a function. Cells, fundamental unit of structure and function. Organelles, the part of the cell that has a specific function. And molecules, a chemical structure consisting of 2 or more units or atoms. Zooming in at ever finer resolution illustrates the principle that underlies reductionism, an approach that reduces complex systems to simpler components that are more manageable to study. Reductionism is a powerful strategy in biology. For example, by studying the molecular structure of DNA that has been extracted from cells, James Watson and Francis Crick inferred the chemical basis of biological inheritance. Despite its importance, reductionism provides an incomplete view of life on Earth, as you'll see next. So reductionism reduces complex systems into simpler components. Emergent properties. Let's re-examine figure 1.3, beginning this time at the molecular level and then zooming out. This approach allows us to see novel properties emerge at each level that are absent from the preceding one. These emergent properties are due to the arrangement and interaction of parts and as complexity increases. For example, although photosynthesis occurs in an intact chloroplast, it will not take place if chlorophyll and other chloroplast molecules are simply mixed in a test tube. The coordinated processes of photosynthesis require a specific organization of these molecules in the chloroplast. Isolated components of living systems, the objects of study in a reductionist approach, lack a number of significant properties that emerge at higher levels of organization. Okay? So for emergent properties, I wrote aspects that emerge with each step higher in the organization of life, which I think is the same one that they have here. No, it's not. I paraphrased it. Emergent properties are not unique to life. A box of bicycle parts won't transport you anywhere, but if they are arranged in a certain way, you can pedal to your chosen destination. Compared with such non-living examples, however, biological systems are far more complex, making the emergent properties of life especially challenging to study. To fully explore emergent properties, biologists today complement reductionism with systems biology, the exploration of biological systems by analyzing the interaction among its parts. In this context, a single leaf cell can be considered a system, as can a frog, an ant colony, or a desert ecosystem. By examining and modeling the dynamic behavior of an integrated network of components, systems biology enables us to pose new kinds of questions. For example, how do networks of molecular interactions in our bodies generate our 24-hour cycle of wakefulness and sleep? At a larger scale, how does a gradual increase in atmospheric carbon dioxide alter ecosystems and the entire biosphere? Systems biology can be used to study all life at all levels. So systems biology is the study of biology that analyzes the interactions among the system's parts. Structure and function. At each level of biological hierarchy, we find a correlation between structure and function. Consider the leaf in figure 1.3. Its broad, flat shape maximizes the capture of sunlight by chloroplasts. Because such correlations of structure and function are common in all living things, analyzing a biological structure gives us clues about what it does and how it works. For example, the hummingbird's anatomy allows its wings to rotate at the shoulder, so hummingbirds have the ability, unique among birds, to fly backwards or hover in space. While hovering, the birds can extend their long, slender beaks into flowers and feed on nectar. The elegant match of form and function in the structures of life is explained by natural selection, which we'll explore shortly. So here I wrote, at each level of biological hierarchy, we find a correlation between structure and function. The cell, an organism's basic unit of structure and function. The cell is the smallest unit of organization that can perform all activities required for life. The so-called cell theory was first developed in the 1800s based on the observations of many scientists. The theory states that all living things are made of cells, which are the basic unit of life. In fact, the actions of organisms are all based on the activities of cells. For instance, the movement of your eyes as you read the sentence results from the activities of muscle and nerve cells. Even a process that occurs on a global scale, such as recycling of carbon atoms, is a product of cellular functions, including the photosynthetic activity of chloroplasts and leaf cells. All cells share certain characteristics. For instance, Every cell is enclosed by a membrane that regulates the passage of materials between the cell and its surroundings. Nevertheless, we distinguish two main forms of cells, prokaryotic and eukaryotic. This is important. Prokaryotic cells are found in two groups of single-cell microorganisms, bacteria and archaea. All other forms of life, including plants and animals, are composed of eukaryotic cells. A eukaryotic cell contains membrane-enclosed organelles. Some organelles, such as the DNA-containing nucleus, are found in the cells of eukaryotes, while other organelles are specific to a particular cell type. For example, the chloroplast in figure 1.3 is an organelle found only in eukaryotic cells that carry out photosynthesis. In contrast to eukaryotic cells, a prokaryotic cell lacks a nucleus or other membrane-enclosed organelles. Furthermore, prokaryotic cells are generally smaller than eukaryotic cells, as shown in figure 1.4. So here in my notes, I wrote, cell theory, 1800s, states that all living organisms are made of cells which determine the actions of organisms. Prokaryotic, single-cell organisms, bacteria and archaea, lacks a nucleus and other membrane-enclosed organelles, smaller. Eukaryotic, multicellular organisms, plants and animals, contains membrane-enclosed organelles that may be specific to particular cell types. And here we're just going to look at this image. So here we see a eukaryotic cell has these little circles, which are the membrane-enclosed organelles. This is the nucleus, I believe. Yes, the nucleus. And let's see, we have the DNA inside of the nucleus. We have the full membrane enclosing the entire cell. We have cytoplasm. And as you can see, these brown spots and orange spots here are membrane-enclosed organelles. And in comparison, the prokaryotic cell here just has DNA. No nucleus, no circles, just a small cell with DNA and a membrane. Okay? Theme. Life's processes involve the expression and transmission of genetic information. Within cells, structures called chromosomes contain genetic information in the form of DNA, deoxyribonucleic acid. In cells that are preparing to divide, the chromosomes may be made visible using a dye that appears blue when bound to DNA. So as you can see in this image, that's DNA as it divides. And you can see it splitting in the second image. Okay. DNA, the genetic material. Each chromosome contains one very long DNA molecule with hundreds or thousands of genes, each a section of the DNA of the chromosome. Transmitted from parents to offspring, genes are the units of inheritance. They encode the information necessary to build all of the molecules synthesized within a cell, which in turn establishes that cell's identity and function. You began as a single cell stocked with DNA inherited from your parents. The replication of that DNA prior to each cell division transmitted copies of the DNA to what eventually became the trillions of cells of your body. As the cells grew and divided, the genetic information encoded by the DNA directed your development. So for my notes here, I wrote, information. Within cells, there are chromosomes that contain genetic information in the form of DNA, chromosomes that contain a DNA molecule has hundreds or thousands of genes. And a gene is a unit of hereditary information found in DNA consisting of a specific nucleotide sequence. We're going to go over what a nucleotide is in just a second. The molecular structure of DNA accounts for its ability to store information. The DNA molecule is made up of two long chains called strands, arranged in a double helix. Each chain is made up of 4 kinds of chemical building blocks called nucleotides, abbreviated A, T, C, and G. Specific sequences of these 4 nucleotides encode the information in genes. The way DNA encodes information is analogous to how we arrange letters of the alphabet into words and phrases with specific meanings. The word rat, for example, evokes a rodent. The words tar and art, which contain the same letters, mean very different things. We can think of nucleotides as a 4-letter alphabet. So here's a DNA molecule here. For many genes, the sequence provides the blueprint for making a protein. For instance, a given bacterial gene may specify a particular protein, such as an enzyme, required to break down a certain sugar molecule, while one particular human gene may denote an enzyme and another gene a different protein, an antibody perhaps, that helps fight off infection. Overall, proteins are major players in building and maintaining the cell and carrying out its activities. Protein encoding genes control protein production indirectly, using a molecule called RNA as an intermediary. The sequence of nucleotides along a gene is transcribed into mRNA, which is then translated into a linked series of protein building blocks called amino acids. Once completed, the amino acid chain forms a specific protein with a unique shape and function. The entire process by which the information in a gene directs the manufacture of a cellular product is called gene expression. Okay. So, I wrote here that for many genes, the sequence provides a blueprint for making a protein. Another gene, another protein. Proteins are major players in building and maintaining the cell and carrying out its activities. I'm just going to scroll down a little bit here. And keep reading. Figure 1.8. Gene expression. Cells use information encoded in a gene to synthesize a functional protein. Excuse me. The lens of an eye, behind the pupil, is able to focus on light because the lens cells are tightly packed with transparent proteins called crystalline. How do lens cells make crystalline proteins? So, a lens cell uses information in DNA to make crystalline proteins. The crystalline gene is a section of DNA in a chromosome. So here it shows you the process from DNA to protein. DNA, part of the crystalline gene. Using the information in the sequence of DNA nucleotides, the cell makes or transcribes a specific RNA molecule called mRNA. So it goes from transcription, DNA, into mRNA. And then from mRNA, the cell translates the information in the sequence of mRNA nucleotides to make a protein, a series of linked amino acids. Okay, so from mRNA, you get a series of linked amino acids, or a chain of amino acid. From the amino acid, you get a chain of amino acid that starts to fold on itself, protein folding. And the chains of the amino acid folds into a specific shape of the crystalline protein. Crystalline proteins can then pack together and focus light, allowing the eye to see. So here I wrote DNA to mRNA during transcription, then during translation, mRNA into a chain of amino acid. And then there's protein folding, where the amino acid folds on itself to become a protein. The result is a protein. In carrying out gene expression, all forms of life employ essentially the same genetic code. A particular sequence of nucleotides means the same thing in one organism as it does in another. Differences between the organisms reflect differences between their nucleotide sequences rather than their genetic codes. The universality of this genetic code is a strong piece of evidence that all life is related. Comparing the sequences in several species for a gene that codes for a particular protein can provide valuable information both about the protein and about the relationship of the species to each other. Molecules of mRNA, like the one in figure 1.8, are translated into proteins, but other RNAs function differently. For example, we have known for decades that some types of RNA are actually components of the cellular machinery that manufactures proteins. In the last few decades, scientists have discovered new classes of RNA that play other roles in the cell, such as regulating the function of protein-coding genes. Genes specify these RNAs as well, and their production is also referred to as gene expression. By carrying the instructions for making proteins in RNAs, and by replicating with each cell division, DNA ensures faithful inheritance of genetic information from generation to generation. So here I wrote, a particular sequence of nucleotides means the same thing in one organism as it does in another. Differences between organisms reflect differences between their nucleotide sequences, rather than between their genetic codes. Some RNAs function differently. Some are components of cellular machinery that manufactures proteins or regulate the function of protein-coding genes. Genomics. Large-scale analysis of DNA sequences. The entire library of genetic instructions that an organism inherits is called a genome. A typical human cell has 2 sets of chromosomes, and each set has approximately 3 billion nucleotide pairs of DNA. If the one-letter abbreviation for the nucleotides of a set were written in the letters the size of those that you are now reading, the genomic text would fill about 700 biology textbooks. Since the early 1990s, the pace at which researchers can determine the sequence of a genome has accelerated at an astounding rate, enabled by the revolution in technology. The genome sequence, the entire sequence of nucleotides, for a representative member of a species, is now known for humans and many other animals, as well as numerous plants, fungi, bacteria, and archaea. To make sense of the deluge of data from genome sequencing projects and the growing catalog of known gene functions, scientists are applying a systems biology approach at the cellular and molecular levels. Rather than investigating a single gene at a time, researchers study whole sets of genes or other DNA in one or more species, an approach called genomics. Likewise, the term proteomics refers to the study of sets of proteins and their properties. The entire set of proteins expressed by a given cell, tissue, or organism is called a proteome. 3 important research developments have made genomic and proteomic approaches possible. One is a high-throughput technology, tools that can analyze many biological samples very rapidly. The second major development is bioinformatics, the use of computational tools to store, organize, and analyze the huge volume of data that results from high-throughput methods. The third development is the formation of interdisciplinary research teams, groups of diverse specialists that may include computer scientists, mathematicians, engineers, chemists, physicists, and of course biologists from a variety of fields. Researchers in such teams aim to learn how the activities of all the proteins and RNAs encoded by the DNA are coordinated in cells and in whole organisms. So for genomics, I wrote, genome is the library of the total of genetic instructions that an organism inherits. A human cell contains 2 similar sets of chromosomes with 3 billion nucleotide pairs in one. The genome sequence. Entire sequence of nucleotides for members of a species is known for humans, animals, plants, fungi, bacteria, and archaea. Systems biology, which is the study of biology that analyzes the interactions among the system's parts at cellular and molecular levels. Genomics, the study of whole sets of genes or other DNA in one or more species. Proteomics, the study of sets of proteins and their functions. Proteome, entire set of proteins expressed by a given cell, tissue, or organism. And important research developments, high throughput technology that provides for rapid analysis of many biological samples. Bioinformatics uses tools to organize, store, and organize the huge volume of data that results from analysis. Formation of interdisciplinary research teams that study how the activities of proteins and RNA are encoded by DNA are coordinated in cells and in whole organisms. Theme: life requires the transfer and transformation of energy and matter. Okay. So energy and matter. Moving, growing, reproducing, and the various cellular activities of life are work, and work requires energy. The input of energy, primarily from the sun, and the transformation of energy from one form to another makes life possible. When a plant's leaves absorb sunlight in the process of photosynthesis, molecules within the leaves convert the energy of sunlight to the chemical energy of food, such as sugars. The chemical energy in the food molecules is then passed along from plants and other photosynthetic organisms, producers, to consumers. A consumer is an organism that feeds on other organisms and their remains. When an organism uses chemical energy to perform work, such as muscle contraction or cell division, some of that energy is lost to the surroundings as heat. As a result, energy flows through an ecosystem in one direction, usually entering as light and exiting as heat. In contrast, chemicals cycle within an ecosystem, where they are used and then recycled. Chemicals that a plant absorbs from the air or soil may be incorporated into the plant's body and then passed to an animal that eats the plant. Eventually, these chemicals will be returned to the environment by decomposers such as bacteria and fungi that break down waste products, leaf litter, and the bodies of dead organisms. The chemicals are then available to be taken up by plants again, thereby completing the cycle. So here, I was very basic with this one. I just wrote, actions require work, moving, growing, reproducing, etc. requires energy. Energy primarily comes from the sun, and the change in energy from one form to another makes life possible. Producers versus consumers, which I think we can all understand. And energy flows through an ecosystem in one direction, entering as light and exiting as heat. And I'm just going to include here chemicals cycle within an ecosystem where they are used and then recycled. Excuse me. Theme. From molecules to ecosystems, interactions are important in biological systems. Interactions. At any level of biological hierarchy, interactions between the components of the systems ensure smooth integration of all parts, such that they function as a whole. This holds true equally well for molecules in a cell and components of an ecosystem. We'll look at both examples. So for molecules, I'll just scroll this up. Molecules within an organism. At lower levels of organization, the interactions between components that make up living organisms, organs, tissues, cells, and molecules, are crucial to their smooth operation. Consider the regulation of blood sugar level, for example. Cells in the body must match the supply of fuel, sugar, to demand, regulating the opposing processes of sugar breakdown and storage. The key is the ability of the biological processes to self-regulate by a mechanism called feedback. Okay? In feedback regulation, the output or product of a process regulates that very process. The most common form of regulation in living systems is negative feedback, a loop in which the response reduces the initial stimulus. As seen in the example of insulin signaling, after a meal, the level of sugar glucose in your blood rises, which stimulates cells of the pancreas to secrete insulin. Insulin in turn causes body cells to take up glucose and liver cells to store it, thus decreasing the blood glucose level. This eliminates the stimulus for insulin secretion, shutting off the pathway. Thus, the output of the process, insulin, negatively regulates that process. Okay? So here I wrote, self-regulation for homeostatic stability is called feedback. Feedback regulation is the regulation of a process by its output or end product. Negative feedback, a loop where the response reduces the initial stimulus. So, insulin and glucose levels, increased blood glucose stimulates cells to secrete insulin, and then insulin circulates throughout the body to decrease glucose levels. Though less common than processes regulated by negative feedback, there are also biological processes regulated by positive feedback, in which an end product speeds up its own production. The clotting of your blood is in response to injury is an example. When a blood vessel is damaged, structures in the blood called platelets begin to aggregate at the site. Positive feedback occurs as chemicals released by the platelets attract more platelets. The platelet pileup then initiates a complex process that seals the wound with a clot. So positive feedback, a loop where the response speeds up the initial stimulus. Blood clotting, platelets aggregate at the site, chemicals attract more platelets, and platelet pileup helps seal up a wound. Okay. Ecosystems. An organism's interactions with other organisms and the physical environment. At the ecosystem level, every organism interacts with other organisms. For instance, an acacia tree interacts with soil microorganisms associated with its roots, insects that live on it, and animals that eat its leaves and fruit. Interactions between organisms include those that are mutually beneficial, as when cleaner fish eat small parasites off a turtle, and those in which one species benefits and the other is harmed, as when a lion kills and eats a zebra. In some interactions between species, both are harmed. For example, when two plants compete for a soil resource that is short in supply. Interactions among organisms help regulate the functioning of the ecosystem as a whole. So, I'm just going to put my notes here. Interactions between organisms can be mutually beneficial, or one species benefits and the other is harmed. Sometimes both species are harmed. Each organism also interacts continuously with physical factors in its environment. The leaves of a tree, for example, absorb light from the sun, take in carbon dioxide from the air, and releases oxygen to the air. The environment is also affected by organisms. For instance, in addition to taking up water and minerals from the soil, the roots of a plant break up rocks as they grow, contributing to the formation of soil. On a global scale, plants and other photosynthetic organisms have generated all of the oxygen in the atmosphere. Like other organisms, we humans interact with our environment. Our interactions sometimes have dire consequences. For example, over the past 150 years, humans have greatly increased the burning of fossil fuels, coal, oil, and gas. This practice releases large amounts of carbon dioxide, CO2, and other gases into the atmosphere, causing heat to be trapped close to Earth's surface. Scientists calculate that the CO2, carbon dioxide, added to the atmosphere by human activities has increased the average temperature of the planet by about 1 degree Celsius since 1900. At the current rates that CO2, carbon dioxide, and other gases are being added to the atmosphere, global models predict an additional rise of at least 3 degrees Celsius before the end of the century. This ongoing global warming is an aspect of climate change, a directional change of the global climate that lasts for 3 decades or more, as opposed to short-term changes in weather. But global warming is not the only way that the climate is changing. Wind and precipitation patterns are also shifting, and extreme weather events such as storms and droughts are occurring more often. Climate change has already affected organisms in their habitats all over the planet. For example, polar bears have lost much of their ice platform from which they hunt, leading to food shortages and increased mortality rates. As habitats deteriorate, hundreds of plants and animal species are shifting their ranges to more suitable locations, but for some, there is insufficient suitable habitat, or they may not be able to migrate quickly enough. As a result, the populations of many species are shrinking in size or even disappearing. So threatened by global warming, this is a lizard, the genus Sceloporus. The loss of population due to the climate change can ultimately result in extinction, the permanent loss of a species. As we explore in greater detail in Concept 56.4, the consequences of these changes for humans and other organisms may be profound. Having considered 4 of the unifying themes, organization, information, energy and matter, and interactions, let's now turn to evolution. There is a consensus among biologists that evolution is the core theme of biology, and it is discussed in detail in the next section. Okay, so this is everything for Concept 1.1. I'm going to go over the last bit of my notes. Sometimes both species are harmed. Interactions among organisms help regulate the functioning of the ecosystem as a whole. Each organism interacts continuously with physical factors in its environment. The environment is also affected by organisms, and humans also interact with our environment. Burning fossil fuels increases carbon dioxide amounts in the atmosphere, thus increasing average temperature of the planet. Global models predict an additional rise of at least 3 degrees Celsius before the end of this century. Climate change, a directional change to the global climate that lasts for 3 decades or more, as opposed to short-term changes in weather. Global warming, wind and precipitation pattern shifting, extreme weather events, storms and droughts are more often, and populations of species shrinking in size or even disappearing due to the changes in environment, leading to extinction. Okay, so we have finished part 1 of this chapter. I will see you guys next time, and next section we'll be going over concept 1.2. Okay, see you guys next time. Bye.